जैसे कि हम बात कर रहे थे कि हमें पीसीआर प्रोडक्ट को प्यूरिफाई करने की ज़रूरत पेश आएगी, बिकॉज इसमें जो अनयूज्ड कंपोनेंट्स रह गए हैं वो हमारे फर्दर एक्सपेरिमेंट के साथ प्रॉब्लम क्रिएट कर सकते हैं। उसके लिए कौन से रिएजेंट्स चाहिए, कौन से बफर्स चाहिए, इसके हवाले से भी बात हो चुकी। अब हम बात करेंगे वो कौन से स्टेप्स हैं जिनको फॉलो करते हुए हम अपना एम्प्लीफाइड डीएनए प्योरीफाई करेंगे। पीसीआर रिएक्शन जब हमने सेटअप किया था उस वक्त एक रिएक्शन की जो वॉल्यूम था दैट वाज 50 माइक्रोलीटर। दिस 50 माइक्रोलीटर वॉल्यूम इज नॉट सफिशिएंट इनफ to proceed further for purification of a sufficient quantity of DNA. Therefore, what we have to do, we have to mix different PCR products of the same group. जैसा कि कंट्रोल, अगर हमने 2 या 3 ट्यूब्स में कंट्रोल का सैंपल एम्पलीफाई किया है तो हम उनको मिक्स कर सकते हैं। अगर हमने पॉजिटिव कंट्रोल को करना है तो हम पॉजिटिव कंट्रोल के अपने रेप्लिकेट्स को मिक्स करेंगे। अगर हम एक्सपेरिमेंटल सैंपल की बात करते हैं तो रिस्पेक्टिव एक्सपेरिमेंटल सैंपल्स जो हैं उनको मिक्सअप करेंगे सो दैट वी कैन हैव सफिशिएंट वॉल्यूम दैट कंटेन्स अराउंड 1 माइक्रोग्राम ऑफ द एम्प्लीफाइड डीएनए। इस एक्सपेरिमेंटल प्रोसीजर में वी आर गोइंग टू पूल डिफरेंट पीसीआर वॉल्यूम्स अप टू 400 माइक्रोलीटर। In the PCR mixture, we have some enzymes and nucleotides. Enzymes are protein by nature. Nucleotides are the nucleic acid or nucleotides by nature. So we have to remove both of these. In the first step, we are going to remove the enzyme. Because enzyme is protein by nature, we will use some enzyme which can degrade this protein. So for that purpose, we are going to use proteinase K along with its buffer. We are going to use a specific volume, 0.2 volumes of buffer containing the enzyme in the 400 microliter of PCR product. Incubate this tube at 60 degrees centigrade. Incubate this mixture at 37 degrees centigrade for 60 minutes. इस पूरे 1 घंटे के प्रोसेस में प्रोटीनेज K is going to act on the enzyme and is going to degrade it. Once this 60 minutes is over, we have to inactivate the proteinase K, and for that purpose it will be heated at 75 degrees centigrade for 20 minutes. Now this is the step when we have inactivated or degraded the protein component or the enzyme in the PCR product. Now we are going to move to precipitate the amplified PCR product and to remove the unused DNTPs. For that purpose, we have to use the phenol chloroform treatment and then chloroform treatment. Once we add chloroform, then we have to go for ammonium acetate treatment in the presence of ethanol. जैसा कि पहले बात हुई, ethanolic ammonium acetate has the ability to dissolve DNTPs. The nucleotides which are present, they will be dissolved in ethanolic ammonium acetate, and for this purpose we have to incubate our reaction mixture for 30 minutes at 4 degrees centigrade. After that, we will see that the amplified DNA has settled at the base of the tube, and the supernatant now contains the ammonium acetate, ethanolic ammonium acetate, along with the dissolved DNTPs. So this supernatant is of no use, so we are going to discard it. The pellet which contains the amplified product, the DNA, is going to be washed with 70% ethanol, and after washing, again we have to centrifuge it so that all the DNA should again settle down, and the 70% ethanol, the supernatant, is no more required. It should be removed. Let this pellet containing the DNA dry, air dry, and then assuming this fact that we are successful in purifying 50 to 80% of the DNA, we have to dissolve this DNA into TE buffer and the volume of TE buffer needs to be calculated, how much volume needs to be used, because we want to have our concentration around 25 nanograms per microliter. So we started with 1 microgram DNA. If we assume that we purified with 50% of the DNA, we have to calculate that how much TE buffer we should add so that our final concentration comes to 25 nanogram per microliter. So up to this stage we have done the purification. Now we have to analyze this purified DNA sample. For this purpose, we are going to use ethidium bromide supplemented agarose gel electrophoresis and along with these DNA markers, so that we can make it sure that the amplified DNA is of correct size and is of correct quantity or the concentration. For that purpose, once the gel electrophoresis is done, we will see the thickness of the band and we will compare it with the shine of the 25 nanogram DNA quantity band, and if there is requirement of adjustment of the volume, that can be done accordingly, so that our purified DNA should have 25 nanogram per microliter concentration.